So hello everybody, welcome on into the first ever stream of the Genetic Society of America. My name is Balint and this is Jessie MacAlpine next to me. We're going to chat you today about some really cool science, including but not limited to Jessie's journey as a science communicator, mycology, the study of mushrooms, and so much more. Jessie, it's great to have you on today.
Yeah, thank you so much for having me. I'm excited to chat about some science.
We've already got some members in the chat as well. We had Golganak here before the stream even started. We have V, we have Grimly, we have Smikes. Hello, everybody. Please, if you have at any time you have questions, feel free to ask all the questions. Jessie is y'all are gonna find out. She's actually a celebrity in Canada in science communication. This is gonna be really, really cool. And Laura Gale, welcome on in as well. So Jessie, do you want to kick us off with just where you are right now, what you're studying?
Yeah, absolutely. So I just finished my PhD in molecular genetics at the University of Toronto. So that basically means that I am a microbiologist. And now when you're a scientist, even after you do your PhD, your journey's not done. So now I'm headed to do a postdoctoral fellowship, which is sort of akin to when a doctor does residency. And I'm going to be doing that at the National Institutes of Health, the NIH in the US. So I'll be moving from my home right now in Toronto, Canada, down to Washington, D.C. in the fall to continue my research. And as Balint alluded to, the area of study that I am in is mycology, medical mycology, and molecular mycology, which basically means that I study fungi. So not the mushrooms we find in the forest, but the tiny microscopic fungal pathogens that can cause disease in humans. So we can launch into all of that. And if people have questions, of course, we can discuss.
Yeah, so what kind of fungus?
Yeah, great question. So there's actually a lot of different types of fungal pathogens that are out there. For me, I specifically study one that's called Candida albicans, which causes some common diseases you might know of, like yeast infections or diaper rash in babies, but it can also cause really, really deadly infection in people who are immunocompromised. So it can cause really severe disease and even death in people who are undergoing, for example, chemotherapy or have recently experienced a transplant. And so we study how these fungi cause disease, why it's so deadly in immunocompromised individuals. And then in my PhD lab, we focused a lot on developing new drugs and new therapies to treat fungal disease. But in the lab I'll be joining from my postdoc, we focus a little more on understanding the host and the human body and how we respond to and fight these pathogens.
So how does one create a new drug for something like, because we're always talking about there's more antibiotic resistance and antifungal resistance coming in, so you presumably have to do drug screening and find new ways to fight off the pathogen. So how does one even do that in a lab setting?
Yeah, that is a fantastic question and one that we wish we had a very simple answer to. I'll talk about two main strategies, but there are others, so this is not strictly comprehensive. The two main strategies are, one, to screen compounds and libraries of compounds. So what that means is different chemical companies, different academic labs have created these giant libraries of compounds, whether that's FDA approved drugs or compounds that have been made but don't have any use yet. And basically researchers can then screen these chemicals using a bunch of different kind of high throughput experiments to find chemicals that already exist that might have activity, in our case, against fungi. And then the second strategy, which is sort of what I focused on, is actually trying to find what we call natural products. So these are chemicals that are already made out in the environment by things like bacteria, fungi, plants. And what we can do is try to identify these chemicals and then exploit them to be used as an antifungal. So in my case, my thesis work actually looked at identifying and describing a chemical that's produced by bacteria that has activity against broad fungal pathogens.
That's really cool. So we already have a question. So with respect to the Candida albicans, is it a comorbidity question versus does it cause death on its own, the fungus?
Yeah, great question. So it definitely can cause death on its own. It's one of those situations of if the fungus ends up in the wrong place, you're not going to be having a good time. So for example, with Candida albicans, it's actually a natural member of the microbial community that lives within our body. So you can find it in mucosal sites like the mouth or the urogenital tract. But if it gets somewhere where it's not supposed to be, such as the bloodstream, it can then cause really deadly infections. Things like sepsis, if you've ever heard of sepsis, which is kind of like a systemic bloodstream infection that has a really high mortality rate. So definitely fungus on its own can cause disease, especially if it gets into the blood or into the brain where it's really not supposed to be.
Is that when it gets into the blood or brain, is there anything that triggers something like that? Or is it stress, or is there an injury? Like, how does that transition happen where you're okay, but all of a sudden it gets really deadly?
Yeah, great question. So for sure, the one thing that makes these infections more deadly is if you're immunocompromised. So if your immune system is not working to the amount you might normally expect. But in terms of how it actually causes these infections, there's all kinds of things. If you get a really deep tissue wound, you could get an infection. Another thing that actually a really cool study that came out in 2020 showed that actually having Candida albicans in your gut, so kind of where all the bacteria and stuff that help us digest our food are, if fungi are in there, that can actually seed these deadly bloodstream infections. If there's any type of injury or if your immune system is not up to scratch, then it can basically seed from the gut into the bloodstream and cause these infections. Now, not something to worry about for the general population, but something that is unfortunately a possibility for people undergoing, for example, chemotherapy.
Wow, so it's again the immunocompromised folks that are at big risk for some of these factors of Candida.
Bingo. Yeah.
One thing that struck me, and we have a comment from the chat. Golganek says, "Congratulations, Dr. MacAlpine, important work."
Thank you.
Absolutely, you're looking on the druggable side of things, and now you're going to figure out how to treat us in the patient side of things as well. One thing I was when you mentioned that you're looking for natural variants as well, things that can treat the infection, right? So I understand how you would go into like a company and you'd order pre-existing molecules and you test for them and see if they work against Candida albicans or not. But for these natural variants, where do you even start?
Yeah, it's a really good question. So for me, I sort of took inspiration from the ecology of these cool microbes that already live within our body. And sort of the one sentence summary of what inspired the work is that in people with vaginas, the leading cause of yeast infections is taking antibiotics. So that led a lot of people to believe that these bacteria that already live in our body are in some way controlling fungus, but we didn't know why or how. So with my PhD work, I wanted to look at these bacteria that were suspected to be able to control fungal growth and then see if we could kind of pinpoint how that was happening. So that doesn't necessarily mean it's a chemical, but it could be a chemical that's doing that. And if that's the case, then that could be a great drug candidate. So starting with that as the inspiration, then what you can do is use a lot of different techniques that we use in biochemistry and in molecular biology to basically take these bacteria, grow them in kind of like a soup, like a rich media that they like to grow in, and then we can basically collect the soup without the bacteria and see what is in there that the bacteria are making that has activity against fungi.
That is really cool. So what is the next step? You have all of a sudden this jar, this soup of potential molecules. Where does one go with that? Like like you had the potential answer in this jar. What do you do next?
Yeah, it's another great question. So basically, it's a process called fractionation. And exactly as the name implies, what we can do is take this soup and kind of break it down to its component parts. So an analogy might be, if you put a bunch of spices into soup and a bunch of ingredients, and instead of making the soup, we're going to try to go in reverse. We're going to take that soup and now try to split it into, the garlic, the ginger, the onions, the water, and see if any of those have activity. And to do that, we can basically use machinery and tools in the labs that can separate the soup based on the chemical properties of its components. And you can basically do iterative fractionation until you get to a simple enough portion of this soup that you can start to identify what's actually in it, because you'll be starting with something with thousands and thousands and thousands of chemicals and then trying to pinpoint it down to one. So it's certainly not an easy or straightforward process.
But so it could be combinations as well. So if you're
Absolutely.
lucky, it might be one and it's the most abundant thing in the soup, but it could be a combination of highly expressed, lowly expressed, and anything in between.
Totally, yeah, and that's what makes this so complicated. And we collaborated because this is not actually the specific area of expertise in the lab that I did my PhD. So that's one of the cool things about being a scientist, is you get to work with all of these other scientists, learn from their expertise to do all of these really complicated experiments, because science basically can't be done in a vacuum. The more people that you collaborate with, usually the more questions that you can answer at the end of the day.
And did you find the molecule?
Yes, we did find the molecule, or at least one of the molecules. We found a single molecule that had activity against a broad number of fungal pathogens. But as in science, it's never the end of the story. So there could, of course, be other small molecules. There could be combinations of things that are effective. But we did identify one small molecule that had activity against fungi.
That's awesome. I can't wait to see you take it to the patient side of things as well in your postdoc work. I think that would be amazing. We have a comment in chat from Laura Gale that her mom passed away of some sepsis due to an infection around the dialysis port. So that's something that you're combating as well by identifying these molecules to hopefully prevent that from happening in the future. We have a fungus fan in the chat. Our friend Smikes asked, how close is this particular fungus you're speaking related phylogenetically to Aspergillus?
Yeah, great question. So, love Aspergillus. Basically, there are many different species of Aspergillus. Some of them can actually cause infection in humans. So, Aspergillus fumigatus is the most common species of Aspergillus that can cause disease in humans. But there are other species within the genera that are kind of found all over, all throughout the environment. And Aspergillus is what's known as a filamentous fungus. So they sort of reproduce and go through their cell cycle in a distinct manner from the yeast fungi. So things like Candida albicans or baker's yeast, Saccharomyces cerevisiae. They are both part of the same larger group of fungi, so they're part of the Ascomycetes, but they are more distantly related than, for example, Candida albicans, my pathogen of interest, and baker's yeast, which are much more closely related.
Cool. So Jessie, let's take it back. Let's take it way back. So you are working on fungus. You're identifying some of the genetic components that could be likely for it to be of how they spread and trying to find out how to fight them. What got you into science? Like let's just start at the beginning. Like what? Because there's a beautiful quote that I saw, I believe on your website, which is by the way pinned for those in the chat and also be linked in the YouTube video. About how you feel that all kids are essentially can be scientists. So how do we get to that? Because that is a beautiful mindset, right? Like if we had more people educate people in that manner and have that mindset, I couldn't imagine the breadth of science that we would have now. So what brought you to that kind of mindset? How did you become the scientist you are today?
Yes, I think I'll start by commenting on that because, this is something I feel really strongly about. I really think that science is a core component of human nature. And I think that's most obvious when you talk to and interact with any young kid. I think the two types of people most excited to be, out in the environment, flipping rocks, picking up salamanders, looking at frogs, are sort of kids and ecology profs. And so I think that one thing I'm very passionate about is that kids have this natural curiosity and desire to explore the world, which in another turn of phrase would be considered science. But somewhere along the way, they kind of lose that and don't recognize maybe necessarily that that is the process of doing science. So I think I can sort of say where I began my scientific journey was by participating in the science fair program in Canada, which is another area that I'm really passionate about, to sort of encourage kids from all walks of life to really take an active participation in the scientific process.
So for me, what is?
Go ahead.
No, There's so many cool things here that us U.S.-based folks might not know. So when you said the science fair in Canada, so when I was a kid growing up, In fourth grade, we had a school science fair. You presented a poster, and that was that. You make it seem like it's much more of a national organization, and so like how does that work, and what is the goal of that? 'Cause that sounds really cool.
Yeah, totally. And I'll blow your mind for a second and say that a similar thing actually exists in the U.S. and the U.S. runs the International Science Fair as well, which I competed in 2013. But essentially, what the Science Fair program is exactly so. Some schools you compete at your school, and then you can make it sort of the regional level, and then in Canada you can make it to either the province. Well, we make it to the province level. In the U.S., you would make it to the state level. So both in Canada and the US, usually participation is grades 7 to 12. In Canada, it's run by Youth Science Canada, which I am on the executive organizing committee for, and it runs the Canada-wide Science Fair, which sees participation from about 500 Canadian students from every single province and territory, so every single corner of Canada you can imagine. And the kids come together once a year with their science projects and compete for all kinds of really cool prizes for a week in a different Canadian city every year. And then similarly in the US, Society for Science runs the Regeneron International Science and Engineering Fair. When I competed, it was the Intel International Science and Engineering Fair. And as Americans, you can make it to the regional level and then to the state level and then on to what's considered your national fair, but the US just kept inviting more and more and more countries until it became the International Science Fair. And I believe now over 70 countries compete in that event, which sees participation from thousands of specifically high school students from all of these countries all over the world. And it rotates between a few different American cities. So I don't know if anyone who's listening has kids or is interested in this, but all of this is basically run by volunteers, volunteer judges, volunteer organizers. So if it's something you're interested in, I can pretty much guarantee that your local science fair is looking for any type of support, whether it be with your time or your money, to sort of continue to support kids being able to pursue science. I know for me, it really completely changed my life because neither of my parents are scientists, but I was really, really interested in science. And I grew up in a smaller town in Canada, so we didn't really have access to a university or any type of sort of extracurricular science activities. But at my school, I saw a poster for the science fair, and I was like, oh, okay, like I'll do a science fair project. And then it sort of just snowballed from there, where I was lucky enough to make it to that Canadian-wide science fair the first year that I participated, and just seeing the other students, the caliber of their research, their excitement for science totally inspired my entire scientific journey. And to this day, going to the Canada-wide Science Fair every year is what continues to motivate me to be a better scientist, seeing how excited these kids are to just do science and ask questions and find answers. That reminds me of kind of why I'm a scientist and why I want to stay in this field and do this for the rest of my life.
That is so cool. So, Jessie, do you by chance remember that science project? That
Oh,
first
of course.
one?
I remember all of them. So my very first science fair project, this was 2008. So think right when An Inconvenient Truth was kind of at its peak. So I wanted to do a project related to the effects of climate change. And I landed on trying to figure out how excessive carbon dioxide, which is one of the greenhouse gases, how that might affect the growth of plants. Because for anyone who might not know, plant biology is really dependent on kind of the atmospheric gases, oxygen, and also these greenhouse gases. So to do that, what I did is I used one of my friend's broken fish tanks, and then I went to Walmart and bought some plants, and I planted the same plants on either side of the fish tank with a glass wall in between. And then I artificially increased the carbon dioxide on one half of the tank and left it the same on the other, and then tried to see what happened. And so I increased CO2 by, if you've ever heard of Alka-Seltzer tablets, which is sort of like what you take, they're like Tums. If you put them in water, they actually emit really, really high levels of carbon dioxide. So in hindsight, I was absolutely like completely poisoning these plants. They were exposed to so much carbon dioxide. But I think I really appreciate the judges at the regional science fair who I think saw the potential and how my thinking aligned. And then sort of by going to the Canada-wide science fair, I didn't win anything, but it certainly like totally sparked me to just carry on and research. And then my research projects kind of went all over the map from there.
So first of all, that's incredible to use those tablets to like, that's so clever to figure out that, it's like an everyday household item, right, that you're able to do science with. So you don't need a fancy lab, you don't need funding to do something that's, if you're driven by your curiosity, you can ask basic questions in a very like at home constrained setting, which I think that's beautifully illustrated by your work. So that's I love that. I was like, well, where is she going with this? How did she increase the CO2? It's like, oh, that is so clever.
I can blow your mind with one of my other science fair projects, because you don't need biosafety to work with Drosophila melanogaster, which for the live stream, they are fruit flies, which are one of the genetic model organisms that scientists use to study basic biology. So we can actually use them to study some of our human biology. So when I was in grade 9, this was in 2009, 2010, I actually, I couldn't go to the lab every day because it was too far away, but a lab at Western University in Canada let me take thousands of fruit flies home. And then my lovely parents let me do genetics experiments from home with my fruit flies. So yes, I did not need a lab, but you definitely need parents who are okay with thousands of fruit flies sort of chilling in your basement.
Did you use FlyNap by chance to put them to sleep?
No, I was again looking at the effects of both excessive carbon dioxide and then cold, so freeze thaw cycles on Drosophila as kind of an understanding of how climate change might impact insect populations. You can see the vein of my interest sort of evolving. Yeah,
so that's the next step, right? So you're going from climate change and plants to the effects in fruit flies. So we got some bugs and some plants going on in here. And so what was the next step? So when moving from high school to undergrad, how did you get interested in the sciences, or I guess continue your interest, right? Because that could be another place where it's just boom, stopped, given if you're in the wrong environment. So like, what did you gravitate towards? Did you join a lab? What were your favorite courses, and how did that foster you into the mycologist that you are today?
Yeah, great question. So I think the journey began with my obsession with microbiology. So I kind of had this fruit fly science fair project, and then the next 2 years I developed a bioherbicide. So basically like a chemical that can be used to kill dandelions or unwanted weeds in agriculture. And then from there, I actually went into malaria research, because previous work had shown that the parasite that causes malaria actually evolved from an ancient algal cell. So previous work had proposed that potentially something that could kill plants could also kill malaria because of this kind of evolutionary history. So I found that fascinating, and that sort of inspired my grade 12 science fair project, which I did the majority of research with at the University of Toronto. So because of that, I then decided to stay at the University of Toronto, or go to the University of Toronto for undergrad. And my whole goal was to do research. So I really enjoyed my courses, but really I was there to do research. That was what brought me the most joy. But I did this kind of specialist program in molecular genetics. And I think what I really enjoyed about that in undergrad was just sort of better understanding the power of molecular biology and molecular genetics to be able to really dissect the reason things are happening. So on a more surface level, what I had observed with my science fair projects is that I could see an effect or see a result, but I couldn't really figure out why that was happening. So for example, I could make this herbicide and it could kill plants, but I still didn't really know why that was happening. And studying genetics and approaching it with genetics and biochemistry sort of let me realize, okay, like I can actually figure out why something is working. And so in undergrad, I joined the lab that I did my PhD in at the University of Toronto, which is where I was exposed to fungal pathogens. And I was immediately sold because what I loved about them is when you study infectious disease, a lot of people think about bacteria or viruses, but they're very different from humans. So when you're trying to kill bacteria, or if you're trying to have some sort of antiviral effect, it's a very different beast to fungi, because fungi are actually more closely related to humans than to plants. So mushrooms being in the vegetable section at the grocery store is actually kind of, it's not biologically accurate. But so what was really interesting to me is that we actually use fungi as a basic model organism for human biology. There's been several Nobel Prizes that have been awarded for scientists that actually studied baker's yeast, the yeast that's in bread and in beer, to make fundamental insights into the biology that governs our own bodies. So that's super cool. But that means that if we then want to kill a fungal pathogen, you're probably going to kill the human host, because they're so closely related. So it's this really, really difficult and interesting challenge where you need to find ways to kill the fungal pathogen that's causing disease without actually impacting the human host and our human biology. And so I found that challenge super interesting, and I obviously have never looked back. Now I'm very much invested in mycology and hopefully plan to stay in medical mycology for the foreseeable future. So.
That's beautiful. So speaking of life, we have a question that's regularly asked. And it was promised it would not be asked, but it is asked. Do you find a virus to be dead or alive? And that's just, again, coming back to like the definition of life is just something that, like, how would you go about defining that a fungus is alive? And in contrast to another organism, like a plant versus bacterium versus virus, like you said, it's more closely related to animals. Does that mean we should look for more signs of life that's more animal-like, or how do you dissect that relationship between, like, in terms of relatedness, and convey that to, let's say, a non-scientist who looks at a fungus and me, and I'm like, I don't see the connection. Like, where do you tie that back together?
Yeah, so actually pretty much the short answer is genetics on that front. So, in the last 100 years of the molecular biology kind of revolution, we've really matured in our ability to study the genes, the instructions that kind of build our bodies and who we are. And lots of incredible scientists and really broad fields have helped accelerate the technology to let us better understand how all elements of life are related via these genes, these instructions in our body, and how they've kind of diverged from each other as we've evolved over time. In terms of viruses being alive, that's also something I don't necessarily want to touch, but I think there are different schools of thought. Obviously, some people think they are alive, some people think they are not alive, some people also think that they are somewhere in the middle, and they may be in the process of becoming alive, for example, as they might develop more sophisticated mechanisms for growth and metabolism. I think there's arguments on both sides. I can highlight the few that I'm aware of briefly, but there's sort of two schools to what makes up life traditionally, and that's that something that wants to pass on its genetic material, and usually some form of something that has metabolism or the ability to grow. Where viruses get murky is that most viruses basically hijack their host to pass on their genetic material. But it's murky because they're still trying to do that. So that's why it's not necessarily a simple answer. As opposed to us as humans, we alone can pass on our genetic material. We're not relying on some host or some other organism to do that for us, if that makes sense.
And so does Canada albicans, back to your current research, does that require the host to grow, or you can grow it externally?
Yep, absolutely. Okay. Yeah. Actually, that's definitely not true, but at least when I'm thinking about some of our single-celled fungi here, most of them can survive well enough on their own as a single cell. What's tricky is there's a big difference between things we can get to grow in the lab and things that are, alive and able to grow out in the environment, and we just haven't figured out how to grow them in the lab well enough yet. But Candida albicans is one that we are very good at growing and have developed mature methods of genetic manipulation and studying of the pathogen, thanks to a whole host of incredible scientists in our field.
Nice. We'll have to get to some of the genetics as well. I wanted to take you back a moment, and how did you pick your undergraduate location? So you mentioned, research was really important to you, and I think we have folks in the stream that are also on the younger side and maybe not like thinking about applying to undergrad, and it's how do you know, because you said, there was these courses and you're there for the research. So how did you know that this is the right place for you in that research setting?
Yeah, that's a good question. And I hope what I say is not too controversial. But my understanding at the time was that U of T had the best research program in Canada. It was certainly at least one of the top universities to pursue graduate studies and to pursue research, especially in medical related fields. And so that was what really drove me to go to the University of Toronto. But because of that, it is also a massive university. So I always try to, when talking to undergrads or giving people advice, I always try to remind people to think about their learning style and their personal learning environment. I was okay in sort of the large learning environment, the massive system of the University of Toronto, but that doesn't negate the incredible undergrad programs at other universities, which might have obviously smaller classes, more hands-on work, more one-on-one experiences, and both can lead to very successful careers in research. I chose to go to the University of Toronto for that specific reason, but that doesn't mean I couldn't have also, come back to U of T for grad school from any other undergraduate university. And for me, I wanted to stay in Canada, so that was also a really big consideration because, of course, there's other great institutions in the US and globally, but for me, I wanted to stay in Canada, so that was an important consideration as well.
Speaking of leaving the US, you said you're going to be going to the NIH, the National Institute
Yes.
of Health, and we have a question from the chat. You talk about your study of fungus for medicinal purposes at the NIH. Do you know if psilocybin,
Psilocybin.
There we go. If that's being studied at the NIH for any psychiatric use, and does that interest you?
Yeah, great question. So I can't comment as to whether or not the NIH is studying psilocybin or if they're studying it related to any type of psychiatric use. I can comment though as to if that interests me. I think as a scientist, as a researcher, I find that very interesting, but it's outside of my realm of expertise. So what Smikes is alluding to is that psilocybin, which is the active ingredient in quote unquote magic mushrooms, which has a variety of psychoactive activity in humans. Some academic research has indicated that microdosing or controlled dosing with this purified substance can have impacts on various psychiatric indications. As far as I'm aware, it's not FDA approved in Canada or the US as of currently. I think that it's about to be approved actually in Australia for certain uses, but I would need to double check that. So someone is welcome to fact check me. But the short answer here is that I think it's very interesting. I think it's a really important area of study, but because I don't study neuroscience, psychiatric disorders, I'm not a psychiatrist, I'm not a clinical professional, I sort of can't comment on being able to head in that direction. But I think like as a human, as a scientist, I think it's cool. And I hope we continue to study obviously anything that can lead to an improvement of some of these psychiatric indications.
Nice.
The Schedule 1 drug in the US. Perfect. Thank you for sharing. Yeah, some of the research has shown promise, I think especially with PTSD.
Nice. So back in your undergrad lab, you're starting in the undergrad lab. You clearly fall in love with it because you end up going to grad school in the same lab. What was your first series of experiments, and what was your first real difficulty with the experiments? Like where, for example, for me, when I was reading once an example of how to wash a sample, it didn't describe to me what washing the sample meant, like pipetting up and down. So I put it under the sink and washed the sample away, but I did wash it. So like what was like, some unique challenges that you felt like you faced, and that now since, you're even more like, you're moving along in career trajectory, like that you look back on now, you're like, that was a great learning experience. Like, I'm glad I messed up on that so I could learn and continue to move forward.
Yeah, so I think mine, I have a story. I guess it's maybe not specifically related to the science, but when I joined my undergraduate lab, the first week of the first summer I joined the lab, this was in my second year of university, I broke my arm while cycling. And so thankfully it was not my dominant hand, and I was convinced I would not be able to work in the lab for the rest of the summer. Thankfully, we were able to work it out with the biosafety officers that I could work, but I had to do everything one-handed as an undergraduate for the first time in this lab. So that definitely really humbled me for obviously, the speed that I was able to accomplish beforehand with both hands, now having to try to rethink sort of how to do all of these tasks with one hand, because I had had quite a bit of research experience leading up to joining this undergraduate lab, but I had always had two hands and sort of not thought about how much that assisted me in the lab. And so that was a really unique and surprising challenge to then be learning all of these techniques and being taught them, and sort of even as I'm teaching them, having to rethink like how I'm going to have to modify for myself. So that's also really helped me recognize sort of ableist rhetoric in the lab as well, because there are so many things that you don't think about until it directly impacts you and your research. So that was an interesting experience that definitely slowed me down for the first few weeks of my undergrad experience.
Can you comment a little bit more about that, like what you mean by, with especially in a lab setting, like the ableist rhetoric, and how do you, now being more aware about it, like what do you do, or how, when you're training someone, what do you take into consideration? Because I think accessibility in the sciences and the STEM fields alone are kind of under talked about, and it's almost like we're not going to chat about it if you don't make us chat about it. So like, how has that experience changed how you view the sciences and how you approach training and these elements to your science?
Yeah, it's a great question. First of all, I should start by saying I'm not an expert in any of these areas. There are much more well-versed advocates and researchers in this field that can better comment on all of the ways in which ableism has a huge detrimental defect or effect on kind of the pursuit of science. But I think that in general, we don't think a lot about how different abilities impact science. So not only physical disabilities, but invisible disabilities as well. And so for me, I think what's tough is obviously when mentoring a student, just being able to sit with them and talk about where they might need specific help. And that could be related to anything. People bring all kinds of different experiences to the lab. So I try to meet my students where they are in terms of their expertise, their interest levels, the types of things that they want to learn in the lab, and whether they have anything that might impact their ability to do that. I think a lot of the physicalities of the lab are also something that are overlooked. So for example, being able to work with two hands, or if you have any type of visual impairment, all of those things can have a significant impact on or are perceived to have an impact. And I think because of that, it discourages potentially some people from pursuing science. And I think that that's such a loss because, as I mentioned, I really think science is a fundamental part of being a human. So there's no reason anyone should be excluded from that process because it's just who we are as people. So anyone who wants to pursue science should be able to, I guess, is my 2 cents.
Beautifully said. And so you're in the lab and you're gaining this expertise. How did you decide to continue that field of study to graduate school?
Yeah, so I think for me, I was really excited about the project. So I had started this cool project where I was looking at these bacteria that live in our body and their effects on these cool fungal pathogens. And the one thing about science is you always think you're done, and you absolutely never are. So I kept thinking, okay, this will be my undergraduate thesis. Okay, that's not possible. Maybe this will be a master's thesis. Okay, that's not possible. Maybe this will be a PhD. Okay, maybe, but it's going to have to be like a couple papers, and then someone else will have to carry on this story. So sort of these things kind of snowball, and it's never a closed book. We're always learning and evolving. And I think to me, that became potentially the most obvious to the public during the COVID-19 pandemic, where you could see in real time the evolution of our understanding of this emerging infection, how it causes disease, how it passes, how we can treat it. That is the process of doing science. We learn a little bit, we learn a little bit more, we adjust, we keep learning, we keep growing, we keep sort of doing the scientific method, asking questions, answering them, and it's always an evolving process. You're always adding to that body of work. And I think that's what's so cool about being a scientist is on the surface, it seems like objectionable facts. You know, you read a science book, this is how the world works, when actually science is the complete opposite of that. As opposed to a body of facts, it's a process. And it's something that we are constantly developing, growing, learning, asking questions. And that's what drives most scientists, not kind of having their list of facts that they can share with people, but like just continuing to learn over and over and over.
It's the chase and being curious and like identifying the next puzzle piece.
Exactly. Yeah.
I will ask you then for a fact. What is your favorite genetic trick that you can do in fungi?
Oh, that's a great question. And that's also a very broad question because, for example, Saccharomyces cerevisiae or baker's yeast is kind of the genetic powerhouse. It's, you know, the foundation of a lot of molecular genetics. But Candida albicans has a so I guess I should, when we do a lot of genetics, you're looking at the mixing of genetic material and doing mutant analysis and things like that. And so for most genetics experiments, this relies on mating in some regards. So whether you're mating yeast or mating flies. But what's really interesting is that in Candida albicans, the human fungal pathogen that I studied, it has a very cryptic sexual cycle. So we basically can't mate it under normal circumstances. So that makes it actually extra challenging to do sort of traditional genetic experiments. And because of that, that means, I don't know if I have a favorite genetic technique because they're all kind of just a little bit harder in Candida albicans than they might be, for example, in Saccharomyces cerevisiae. But I think my one sentence summary of what I like the most in genetics is I'm a big fan of conditional expression mutants. So this means that you can take a gene of interest and you can kind of modify how it's controlled. So if I'm interested in a gene that does something in a cell, I can then modify how it's controlled. So I can see what happens maybe if I have too much of that gene product, maybe if I have too little of that gene product. And it's a very, very powerful genetic technique because it can allow us to study genes that are really important for cell growth or might have different effects on the cell. And if none of this makes sense, please ask clarifying questions. But that's one of my favorite things to do when making a genetic mutant.
You caught my interest at another point. Why is there trouble having them mate the Candida albicans strains? What about their biology makes that difficult?
Well, so basically we can't find. Now this is like a super ongoing area of research. So you know, someone could watch this YouTube back in a couple years and be like, wait a second, we know all these things. So based on our current knowledge, we can't really get Candida albicans to easily mate. There is a way to get it to mate, but it doesn't seem to happen very commonly or under natural circumstances, kind of everyday circumstances in the lab or in the environment. What Candida albicans does instead is it's very, very amenable to what we call aneuploidies, which means just having the wrong number of chromosomes. Many organisms don't want this because it can be super sick and have a lot of effects on the cell. But in Candida albicans, it can really handle different numbers of chromosomes. And some people think that's how it can generate the genetic diversity that you need to be able to evolve and adapt to changing environments. But the short answer is that for the most part, when you think about evolutionary history, sex or traditional mating is something that is really, really beneficial to organisms and in many ways helps sort of push things along as multicellular organisms evolve. So it's unclear why actually many of these human fungal pathogens have these kind of cryptic sexual cycles, which means they don't seem to kind of readily undergo it. They need to be kind of coaxed into it in very specific and environments that seem unclear to what they might be exposed to in the natural environment.
That would be really cool to figure out why that is, like you said, in a couple of years of like.
I know people who are working on it, so I'm sure that they are well on their way to getting us some better answers.
It's weird. It's like there's several organisms that you just can't mate in a lab, and it really makes it difficult to ask those other side of fundamental questions you really want to go for. And so it'll be really cool to see in a couple of years what the update is. We'll have to bring you back on, Jessie. You have to tell us what happened. We do have
and I should be clear, they can mate. It's just under very cryptic circumstances. So it means when we're making genetic mutants, for many people, that's not the fastest way to get to the mutant that you want to study. So you use more traditional genetic techniques.
Gotcha. We have a first-time chat from Maddie B, and their comment along with Smike's are a little bit similar. So Smike says, seems like individuals identifying as female are still poorly represented in some areas of the hard science. Making assumptions, and I apologize, but how has that affected your journey, and was there a mentor that you particularly looked up to?
I actually love this question because I've talked about the science fair program a little bit. And what I love the most about the science fair program is that we actually have more participation by students who identify as women than we do by students who identify as other genders. And so I think what to me was really interesting and surprising by that is when I was competing in the science fair, I'd actually often get asked this question by, media sources or people interviewing, and I was always so confused. I was like, well, what do you mean? Because all of my friends who are scientists are girls. Like, I didn't understand it was a problem competing in the science fair. And I think what's crazy and very interesting is kind of the further you get in academia, the more that becomes a bit more obvious. So I'm a biologist, which I think has made some of the most progress in this area. So in my undergraduate program, it was again, majority people who identify as women. And then in grad school, I would say maybe it was a bit closer to 50-50. And postdoc, we'll see, I'm headed there now. And I think for many departments, looking at the faculty level, it still remains majority people who identify as men. And this is as someone, like as a white woman who I think, there are many examples of white female scientists that I'm able to look up to, whereas other minority groups don't necessarily have people in faculty positions to be able to look up to due to all types of systemic reasons that have impacted who ends up in those faculty positions. So for me, now I feel like it's had a bigger impact on my identity as a scientist than as a kid. But because I had those early experiences, I never had that assumption. It never felt, and I was really lucky about this, it never felt like I couldn't be a scientist, because looking around me at the science fair, all of my friends were people who identify as women. So I was sort of like, well, we're all here, so science must be like what we want to do. And then it's really unfortunately been people in positions of power who have disappointed me throughout my academic career sort of in this regard. And hopefully that's maybe the best way of answering that without pushing too many buttons.
I think that's a fair way definitely to answer it. Maddie was wondering, do you have a scientist that inspired you the most?
That is a really good question. So a scientist, I think a lot of my early science fair mentors definitely had a big impact on me as a scientist. So the lab at Western University that assisted me with those early fruit fly experiments, that was Dr. Brent Sinclair and his graduate student at the time, Dr. Katie Marshall, who's now a professor at the University of British Columbia. And both of them were hugely instrumental in my early interest in genetics, in biology, in the pursuit of academic science, because both of them were never fazed by my age or my lack of experience, and they were always open and welcoming to answer any question, to provide any feedback, to really walk me through what it's actually like being a scientist. And having that early experience really helped to encourage me through some of kind of the grunt work of maybe traditional school science that was not reflective of what I was seeing in the lab and these two amazing scientists that I got to interact with. On a, I guess, more recent front, my current the supervisor of my PhD, who I'll be leaving, Dr. Leah Cowan, and a previous postdoc in the lab, Dr. Teresa O'Mara, both have been really, really important mentors and very inspirational to my career path, especially when it comes to medical mycology. Both have been really important for helping me to grow both as a scientist and as a person. And I really look up to both of them in all aspects of my life. I still sort of live by the motto, what would Teresa do whenever I hit a problem in the lab? She was my first mentor when I joined an undergrad and had to deal with my broken arm.
Speaking of mentors, I would argue you've become a giant mentor across Canada. Could you tell us about some of your SciComm efforts that you've driven, everything from AMAs to TV shows to photography to just, there's so much that you have done. And it is just, I just want to highlight it to everyone about everything that you've done. Like, maybe let's start with like the TV show. Like, please tell us about that.
Yeah, so I think one thing that really drives me, as you might have noticed, is that I really want every kid to be able to realistically imagine that they could be a scientist. If that's not something they're interested in, no problem, but I want everyone to have that choice to really decide it for themselves. So I was really lucky that I got the opportunity to sort of fact check, help to write, and then ultimately act in a children's television show that is in Canada. So unfortunately, because it's sponsored by the Canadian government, it's not available to watch in the US. But it's this really cool kids TV show that's called Blink and Azoo. It is on YouTube if you're based in Canada or have a VPN. And it's meant for young children, but the premise of the show is that a kid will ask a kid type question. So, how can I stay up all night? How can I make my vegetables taste better? How can I meet a dinosaur? How can I grow a new arm? And then what'll happen is kid will ask that question, and then a scientist will come in and answer that to the best of their ability. And what I really like about the show is I'm not the only scientist that acts on the show. There are a variety of other actors from every walk of life. And the point was that any kid should be able to watch the show and see themselves reflected in that scientist, in that expert who answers that question. And I think I also like that it really started with a kid question. Like one of the episodes is, why is poop brown? Like the type of questions that kids ask, and then kind of shows how science can help us answer some of those questions. So I've really enjoyed being a part of that. And we have 2 seasons, and I think, maybe there'll be more, unclear yet, but so far, that's been something that I've been really proud of to be able to help to shape, at least for Canadian youth for now.
What was your favorite question so far on the TV show that you answered?
Well, my favorite question, which is the most watched episode, of course relates to fungi, and ostensibly it began with the question, why is my bread moldy? And then went on a journey from there about fungal biology. So
nice.
Yeah.
We have I think almost an evergreen question at this point is from Smikes. Did you ever run into the unfortunate attitude that real scientists, quote unquote, don't do SciComm? And then how do you think that this perspective can, by the old guard, can be changed?
Yeah, this is really tough, I think, is sort of the short answer. I think, unfortunately, there is an attitude in some circles that unless all of your time is completely dedicated to your research, then that means that you are not a successful scientist. And I think what's frustrating there is that everyone has hobbies, everyone has things that they do outside of the lab. And as scientists, I think one of our responsibilities is sharing what we do and what we find with the public. So I have not personally experienced this yet. But I know of many of my colleagues who have experienced this type of attitude. And I think it's something that's really frustrating and very much related back to that there is one type of scientist and one pathway to success in the sciences, which is just not the case and very much not true. And I think that the more perspectives that we bring into science, the better it will be. What I learned the most in the science fair is that I would get some of the best questions from the kids who were doing nothing related to my project. And that would completely shift my perspective on a challenge and often shift the direction of my research. So if we are unwilling or unable to talk about our science with people outside of our field or outside of our expertise, then it'll be hard to sort of change those perspectives and continue to welcome a diversity of voices within science, which I think is just so important to the process of pushing science forward. So hopefully I won't come up against this in the future. And I really hope that it can change as people realize the importance of science communication. And once again, I refer back to that I think the COVID-19 pandemic really highlighted how important it is to be constantly talking about the scientific process, being a scientist, and the process of research, not just when it's a global pandemic, but at all times.
And so one way that I know that you've wanted to attack talking more about science and like making a difference as well as you joined the Genetic Society of America Early Career Leadership Program. So he goes, why, like, what was your mission there? Why did you join? And like, what are you doing there to promote this idea as well of like pushing more science to a broader community?
Yeah, so one of the reasons I'm here today is that I recently became co-chair of the Early Career Leadership Program Communication and Outreach Subcommittee at the Genetic Society of America. One of the reasons that I wanted to do that is a lot of my personal science communication efforts up until today have been really intertwined with the science fair, which has been fantastic, but is for sort of a certain audience of people and people who are really interested in sort of doing science themselves. One thing I hoped with joining the ECLP program is that I could try to start to reach people that are maybe interested in science and interested in learning more about what scientists do without necessarily wanting to do it themselves, because I think that's a really important population that we should be continuing to reach out to, because science is cool. And I know I say that with some bias, but it is pretty fun. And I think that there's unique opportunities to kind of showcase that to people, even if they don't necessarily want to do the experiments themselves. Just like I might be interested in, a certain career on the surface without actually wanting to do it myself. And I think that was one of the things that I wanted to do with the GSA, and then just to reach broader voices, as I've been pretty limited with sort of the Canadian focus. And I think the cool thing about the GSA is they connect this incredible community of genetics researchers globally. So you can reach this broader audience of people who might be interested in genetics because they're really fun.
Smikes is quoting one that we say is science is like magic but real. It's just cool and always exciting to be around.
Totally.
What project are you most excited about that you're working on now?
At the GSA or my current research?
Both.
Okay, so I think at the GSA, we've been talking about this for many months now, but I think it will finally begin. Is we're going to be working on a more concerted social media effort from our subcommittee, where through the genetics, the GSA Instagram account, which you're all welcome to follow, we'll be posting hopefully cool little tidbits about interesting areas of biology. So one example that I like to use is there's actually a gene in fruit flies that's named after Sonic the Hedgehog, and there's a whole interesting story around why that exists. And yes, Sonic the Hedgehog, like the video game character. So try to kind of create posts where we can connect these cool science ideas and topical things in genetics with maybe something that might be interesting to everyone, of, how do we come up with gene names, and how does one end up named after a video game character? So narratives like that. And then from my current research, I unfortunately can't comment on the project that I'm most excited about, but what I can say is that we're continuing to study this chemical that I discovered and sort of seeing that it has broad activity across many fungal pathogens, and every new result that comes out is exciting. So I'm excited to see where that'll head and how I'll be able to continue to translate that in the next few years.
So, Jessie, speaking of video games, you did a Last of Us AMA, the Ask Me Anything style Last of Us, the video game. I will link it into the chat now. We'll also link it on the YouTube page once this goes live. We can watch parts of it, but I just wanted to ask you, why The Last of Us? And, for if I'm just sitting here like, what science is there in a PS3 video game? Just convince us why that's what the science is there and why it's based on real things, why we shouldn't be scared that we're not all going to turn into zombies. Tell us about it. What was that, and what was that experience like as well?
Yeah, so I think for anyone who doesn't know, The Last of Us is obviously a video game, part 1 and part 2, and then recently HBO, I think in February or early in the year, came out with a dramatic adaptation of the video game that recently got nominated for several Emmys. And I think what really excited basically the entire fungal field is that kind of the common thing for every fungal researcher is that fungal pathogens are famously overlooked. People know about yeast infections, people know about diaper rash, and then it's kind of left at that because people sort of don't necessarily worry about fungi until you become immunocompromised, and then all of a sudden there are these crazy deadly infections, and we have only a few drugs that can work against those infections. So with bacterial infections, we have dozens of types of drugs. With fungi, there are only 3, and really only 2 because the third is so toxic. So it's a huge, huge problem in the field, and yet no one seems to sort of, or let's say maybe the public awareness of that is limited. So what was really exciting about The Last of Us is for, anyone who played the game, anyone who watched the show, it was one of the most realistic interpretations of a zombie that's happened. Now, that doesn't mean it's scientifically accurate. It just means it was a really unique take on the process of someone becoming a zombie that had a kernel of truth that I think made it a little bit more unsettling. It made the game extra unique, and then it obviously created this really interesting artistic interpretation in the television show as well. And so we can get into that, but in the AMA, I sort of talk about what is the science behind this zombie fungus? Is this at all possible? Do we have any examples of fungi kind of mass manipulating human behavior, things like that? And so as a mycologist, what was great as the show was coming out is my whole community of colleagues, all over Twitter. It was friends being interviewed and people I know being experts in articles and things like that. And so it's just been an interesting way to kind of raise awareness about fungal pathogens and hopefully get people excited about studying these super cool diseases.
So what are those 3 deadly fungi? And you said the third one's
3?
what? And what's the crazy deadly one?
It is groups of 3. So 3 is actually the antifungals. So there are 3 main classes of drugs that we have to treat fungal infections. And they're the pollines, the azoles, and the echinocandins. But there actually are 3 main genera of fungal pathogens that cause disease, and that's the Candida species. I studied Candida albicans, the Aspergillus species, which we talked about a little bit earlier, and then the Cryptococcus species, which are Basidiomycetes. So they are very divergent from Candida and Aspergillus. But there's about 300 fungi that are capable of infecting and causing disease in humans, and that number is expected to grow with climate change.
Why?
Great question. So actually that's to a certain degree implied actually in the origins of The Last of Us. So there's a theory in the fungal field, which is really cool, that has been sort of pioneered by Arturo Casadevall at Johns Hopkins. And that theory is that human body temperature specifically evolved to combat fungal pathogens. And here's why. There's nowhere to prove this, but this is the theory. Human body temperature is about 37 degrees Celsius. That's actually the optimal temperature for bacterial growth. When we grow bacteria in the lab, we grow them at 37. So that temperature is not really doing anything to bacteria, unless you get a fever and kind of pump your body temperature up a little bit. But fungi prefer to grow at 30. When we grow fungi in the lab, when we find them in the environment, they're usually growing at 30 degrees Celsius or lower. So for most fungi, when we breathe them in, when we eat them in our food, they hit that 37 and they die. They can't survive. What is the concern with climate change is that as global temperatures continue to rise, can there be more crossover of microscopic fungi that are capable of surviving at human body temperature and causing disease, like some of these other fungal pathogens? And there's been an emergence of a fungal pathogen since 2009, Candida auris, which has caused great concern globally, and incidences of it increased during the pandemic. And one proposal of how it emerged is that potentially due to rising temperatures from climate change, it emerged, but that has not been convincingly showed yet. It remains sort of a hypothesis, and even environmental sources of Candida auris remain relatively enigmatic.
So, just to get back to The Last of Us, can any of them that you just described, mind control us?
Yeah, it's a great question. So none of the fungi that infect humans are capable of what is shown in the video game or the TV show, is sort of the short answer. But what the show is sort of inspired by is this type of fungi called Ophiocordyceps. But in the show, they talk about it as cordyceps, which is technically a different fungus. But what this fungus is capable of doing is it actually infects insects, and in particular things like ants. And what it will do is it will take over the vast majority of the body mass of that insect. And over time, it'll modulate its behavior. So it begins by sort of altering, when the insect might be active. So if it's normally nocturnal, it might be active during the day or things like that. It'll also start to kind of dissociate from its community. For example, as an ant, it might not interact with the colony to the same degree until it reaches a point where it will basically climb to a really high, tree, branch, something like that. It'll clamp onto a branch or whatever it's climbed to, and then it will die. And the fungus will sprout out of the insect's body to spread its spores across the forest. And so it's called the zombie fungus because it does alter the behavior of these insects and ultimately kill them in service of spreading its own spores as widely as possible. Now, that has been taken to obviously an autistic interpretation in The Last of Us, but because that does happen in the natural environment, some people have seen that as a slightly more realistic zombie origin than sometimes, it's sort of like a mystical, magical origin that there's sort of like no basis to at all. But this does occur with insects. The caveat here being that Ophiocordyceps and insects have sort of co-evolved for millions and millions of years. And for that to occur in a human, nothing is impossible, but it would be very, very beyond the realm of reasonable possibility in sort of the time frame that is implied in the television show. But still kind of interesting to think about, especially because we can have a fungal pandemic. It just might not necessarily cause these sort of bloodthirsty zombies, but a fungal pandemic is still something that can be of concern.
So speaking of, we had a follow from Debs Barreto, and we have also Dr. Waleed, who is another member of the ECLP, saying hello for the first time Twitch stream. Welcome in, y'all. So, us eating each other as zombies probably won't happen as fungi, like fungal spreading, at least not in the near future. What kind of big pandemic zombies, like, not zombie level, like not zombie, but like zombie level spread, are you worried about that could in fact happen? In terms of like, you're having the increase in climate change temperatures, like what is a public health threat, like in your studies, that you'd be worried about?
I'll make a one sentence joke here, which I think the biggest threat to public health is humans themselves. I think that unfortunately sometimes that can get in the way of successful pandemic responsiveness. I think that for me personally, so this will be a personal opinion, not reflective of obviously the entire field, I think in considering a fungal pandemic, fungal pathogens are particularly bad at human to human transmission. So I think that level of a pandemic could be unlikely. But what might be more likely is exactly some sort of shift in the environment that could result in massive exposure of a human population to a given fungal pathogen without necessarily needing that human to human transmission that we see with something like COVID, for example. But I do think at the end of the day, there's a reason some of the most frightening pandemics have been viruses. And I think that's because we really do have very limited therapeutics against viruses. Antibiotics can treat bacteria, or I should say antibacterial agents can treat bacteria. We have a few antifungal treatments that can treat fungi, but we are really limited when it comes to our arsenal of antiviral treatment options. And viruses, due to their size and their nature of their replication, can evolve very, very rapidly, as you might have seen with the COVID-19 pandemic, where we have the emergence of these variants. And so I constantly remain awake at night thinking about what the next viral pandemic could be. But I also think what's been one really important thing we've learned from the pandemic is how some very simple tools can be really effective against a broad range of illnesses, for example, masks. And so I think that has also been really reassuring to know, even what might emerge next, we do have some tools in our arsenal that can be really effective and aren't necessarily specific to any given infection. They'd be effective against bacteria, fungi, or viruses. So.
It's a really well thought out and good answer, Jessie. I like that.
Okay.
With respect to the fungal growth and the cordyceps that you were talking about, there's also we've seen other fungi that look like, studied in the lab. I know it's all over Twitter. It's particular, like slime molds are said to have like a mind of their own and how they move. And so is there any likelihood that the story of The Last of Us was at all involved, like with bringing in both cordyceps and slime molds? And is that at all biologically accurate?
Yeah, so this is one thing I loved about The Last of Us as a mycologist, which was that a lot of the visuals that you see are really interpretations of very diverse fungal organisms. So for example, in the title sequence, and one scene in the show, this shouldn't be a spoiler alert, but I guess if you haven't seen the show, maybe pause. But at one scene, you see someone who's been infected with the fungus, and they're sort of up against a wall, and they have sort of fungi growing out of their body, and then what appears like a slime mold growing kind of all around them. And what's really interesting to me about that is these are very diverse fungi. This is not reflective of Ophiocordyceps biology or cordyceps biology. It's sort of taken really interesting artistic elements of different, very beautiful fungi. So slime molds actually aren't even fungi. They're technically in the animal kingdom. They are basically like closer to a single-celled animal organism as opposed to a fungal organism. But they're super cool looking and are beautiful, and there's a reason they were considered fungi for so long, because they look like a mold. So as a mycologist, it's kind of interesting to watch that, to see like a very stunning visual interpretation of what a fungal pandemic could be, not necessarily reflective of a single species infecting the human body. But it's cool because really, like, there haven't been that many interesting artistic or interpretations of fungal pathogens in the media in general. So it's still really exciting to kind of see how they chose to represent different elements. Even the spreading of the spores in the video game, the characters need to wear masks so that they don't breathe in the fungal spores. And as far as I'm aware, it was an active choice in the television show to kind of not mention that in the narrative, as otherwise the whole series would be the characters in masks. And we've already had Pedro Pascal in a show where he wore a mask the entire time. So maybe they were hoping to see that human-human interaction a little bit closer.
Well, he's helping another kid, right? So that's his theme.
Yeah,
right?
Exactly. Yeah.
So as a mycologist, then would you say there's been benefit for The Last of Us and these kinds of like pop sci representations for the field, or are you on the side where it's like negatives? Like, and how do you gauge something like that? Because I know it can drive people crazy when you hear about CRISPR and there's these movies where you're becoming a lizard man, right? And it's like, it might introduce the idea of it, but it goes so far left field of what the science is that you're doing a lot of backtracking to to explain what you actually can do. Have you had that feeling with The Last of Us as a mycologist, or has it been more of a people are just now excited about it and it's even easier to talk about your research?
Yeah, I actually think it's a little bit of the later. So I think what I love about the mycology community is that we are, in my opinion, like a really wonderful, really welcoming community that's just excited to talk about fungi. And so for a lot of my colleagues, they've sort of seen The Last of Us as a great jumping off point to sort of say, yeah, this show is super cool and fungi are wild. And here's some of the really cool things that we know and we don't know. And I think my interpretation is that that has been the approach as opposed to kind of, oh, The Last of Us is, all incorrect and here's all the ways that it's wrong. I think it's a cool way to get people excited about fungi, and I think that's where it's been really successful, at least for us as mycologists. So I'm certainly not upset by it. There's a little bit of backtracking, but I think even as the public, you recognize things are going to be taken to an extreme when it comes to zombies. But I agree that this could be harder when thinking about things like genetic mutants. I think sometimes that's in such a shrine of concern that can take a bit more effort to backpedal from, versus zombies are sort of already seen as an extreme. So the backpedaling feels a little bit more easy to do.
Laura Gale says my mom once tried growing mushrooms in the basement and then not realizing that the spores would get in the air and the vents and grow in there as well. So that's another, I guess, example of how The Last of Us did get it right from the mycologist side of things, of how the spores are dispersed.
Yeah. Although I think that they really overlooked the fact that if humans breathed them in, they would get infected. But I get why they avoided that. But yeah, most fungi reproduce via the production of these spores. So that's how many of these environmental fungal pathogens can start as a lung disease and then disseminate, or spread throughout our homes and kind of grow fungus everywhere.
So when you're growing them in the lab, what do you have to do? Like, how do you prevent yourself from getting sick?
Yeah, super great question. So when we were talking about Aspergillus, I kind of talked about the two main types of fungus that we work in the lab. We work with these yeast type fungus and then filamentous fungus. So yeast type fungus are things like I work with, Candida albicans and Saccharomyces cerevisiae. They don't produce these airborne spores in the lab that we necessarily need to worry about, versus Aspergillus and other filamentous fungi do. So if I'm working with an Aspergillus culture that's mature and I go to scrape up the spores, I can literally see little poofs of spores being produced. So because of that, we work in a biosafety cabinet in accordance with all of the Canadian laws that are in place to keep both us and the community safe. What's ironic is that when it comes to Aspergillus, there's definitely way more Aspergillus in the air we're breathing outside than even with what I'm working with in the lab. It's a totally ubiquitous environmental fungus, which is not a problem for anyone with a strong immune system, but it can be a concern if you're immunocompromised. So in the lab, I wear my lab coat, I work in a biosafety cabinet, gloves, washing hands, all the usual PPE. That's all required by the Canadian government to keep us safe.
Nice. Speaking of beautiful fungi, we're going to switch to a different scene. And y'all, you're going to like this. Jessie is a professional photographer of fungi.
Oh, professional is a strong word. Let's say hobbyist.
Hobbyist, but amazing photographer. And so it's a pinned message to her website that you can check out and see all these photos. And I thought it would just be kind of cool for us to show off all these unique fungi that you photographed. And if there's anything...
need to update it, though, I should say. I'll add some more of my recent finds.
Guys, bookmark the page so you can check it out, the updates. So can you just tell us about just by eye, if we're looking at these, they look very different. And what differences are here that are meaningful? Like, is there reproductive differences? Are there toxicity? Like, how do you know, just by looking at this, any fundamental things about these mushrooms? And there's a lot here that we can talk about, but whichever ones that you feel like are really interesting, could you tell us about them and why you think they're cool?
Yeah, so I think a couple cool places to start here. So if you're on my website, I don't have any other blogs posted yet, but I do have one blog, which is my tips for getting started with fungal identification and just getting used to seeing mushrooms in the woods. And I always like to start with saying, when we picture cool stuff in the wild, you often picture like the rainforest, right? That's where you get many of the coolest insects, coolest plants, coolest animals. But when it comes to fungi, Canada and the northern US and the coast has some of the most incredible fungal diversity in the world. If you're looking to getting into spotting really cool diversity within a kingdom of life, fungi are actually one of the best places to start in North America, is my recommendation to everyone listening. So for me, I use a couple different tools, which I have outlined in my blog post on the website. But one of the main ones is actually just a free app that you can get on your smartphone called iNaturalist. And with that, what you can do is take pictures of fungi that you find out in the wild. And then with that, it will suggest the genera that it thinks it might be based on your geographical location. And then I usually just sort of cross-reference that with Google Images. And that's how I've begun to start to build up recognition of different common fungi within where I live, which is kind of southwestern Ontario. So it's a free tool, and what's really cool is scientists then go on this app and they use that as some of their crowdsourcing data for where certain species have been identified, and experts can go in and comment on your identifications to confirm whether they're accurate or not. So if I find a fungus in the forest, I think I know what it is, I post that on iNaturalist. Experts can then come and say, yep, I agree with this ID, or no, here's my slight suggestion as to what it might be. So it's been a really great learning tool as I learned to identify mushrooms, because even though I'm a mycologist, I really focus on the microscopic mushrooms. So I'm still learning when it comes to sort of the classical mushrooms that we see out in the forest. So yeah, on my photography page are just some of the mushrooms that I've found out and about, sort of in southwestern Ontario. None of these are from exotic locations. Most are from just your everyday neighborhoods. And actually you see the right hand photo there, that really beautiful sort of orange mushroom, that was actually found next to a parking lot. So
really?
sometimes the coolest fungi can be found in the most unexciting locations, ostensibly. Backyards are a great place, anywhere that's kind of lush, moist. This is actually a great time of year to be spotting, between kind of now and November. There should be lots of stuff out there. But yeah, a couple of my favorites are on here. Maybe we can scroll for a second and I'll highlight a few that I've really liked. Okay, yeah, so if we pause here, on the right-hand side here, that fungus is colloquially referred to as old man's beard, because as you might be able to tell, yeah, that's it grows on the sides of trees, and it looks like a beard. And so these are actually very common in Ontario, in southern Canada and the northern US. They're beautiful to see out in the wild. They are edible, but my number one takeaway here is never to touch, eat, or collect any mushroom that you are not 100% certain is edible. And I wouldn't recommend ever collecting anything without expert input. And that's actually the advice I use for myself as well. So I did not collect or eat this old man's beard, and I don't actually regularly eat or collect anything that I find. I just like to go out and find and spot and identify mushrooms, and that's still a really fun activity.
We have some folks in the chat who suggest it looks nothing like their beard.
Oh, yes, yeah.
No, that
maybe not yet. I don't know your ages, but maybe in time.
Not yet. So when you were saying, we'll keep scrolling. I was just curious, like, that now is a good time to go mushroom spotting through November.
Yep.
So what about this time of the year makes it ideal for mushroom growth? And does that also correspond to an increase in the likelihood of someone contracting a fungal disease like this time of year? Or is that totally separate?
Yeah, that's a great question. So yes and no. There are obviously some skin fungal infections that sort of thrive in kind of moist environments, and that can often increase in the summer months. So if you think of things like diaper rash with babies. That can often increase sometimes depending on kind of moisture levels of the skin and being sweaty. And there are some skin fungal pathogens associated with that. So that might increase in the summer. But as far as I am aware, again, I am not a clinician, so this is just based on my understanding of the literature. I don't think that necessarily, a lot of these more deadly fungal infections are much more closely associated with your immune status than any sort of cycles of the environment. Although environmental exposure can obviously be one of the ways that you get those infections as someone who's immunocompromised. In terms of the cycles of fungi, what I actually like to think about as mushrooms is I think that they will emerge in cycles as well. So there are types of mushrooms you're more likely to find in the spring, one's more likely in the summer, one's more likely in the fall. So really you can find them anytime where there's not snow covering the ground. Sometimes it gets harder in the late fall if there's kind of leaves covering everything. That can make it a little bit tougher as well. But there are cycles to everything. One reason I find this time of year particularly good is usually you're getting, sort of massive rainfalls. Forests are starting to become a little bit more lush. That moisture really helps fungi to thrive. As sort of material from the winter is decaying, that's as the mushrooms are thriving. And we have slightly warmer temperatures, which can also support certain mushrooms to grow.
And you would get these kinds of questions too. Smikes is wondering, because you're our expert now, right, on all things mushroom.
Yeah.
So Smikes, when I was a child, we had these fungi-looking pods, northeast New England, that we used to stomp on, and fine yellow dust would be released. Any idea what those were?
Yeah, I think so. So were they round? Were they like round kind of balls that you would poke and the spores would come out?
Yep.
Yeah, okay. So totally, those are super common. And as you are poking them, that's the spores that are being released. So actually, do I have any on the site? Let's see here.
Tell me when to stop.
They would be round. I might not have any. Oh, no, that's a coral. But they were probably like earth balls maybe, or something in that realm. Actually, I wonder if, I don't want to show you a screen on a screen, but I actually think I probably have a video I can show you from last fall, and you can tell me if this looks like what you saw. Let's see here. Sorry everyone for this, but hopefully could be interesting.
Then while you're looking,
these ones are not mature yet, but were they sort of shaped like this? Oh yeah, so they weren't necessarily puff balls. They might have been earth balls. Puff balls are often quite bigger, but no, you were not too close to dying. Thankfully, those are honestly, the deadliest mushrooms are they're the angel of death and related mushrooms, and they usually grow as a single white mushroom that has a veil on the stipe, which is kind of like the stem. Always avoid even touching. I would avoid touching fungi in general if you're unsure what they are, but specifically sort of lone white mushrooms are definitely one of the biggest things to worry about.
So these,
of course,
on screen,
they're also very common. Yeah, so they're one of them. There's the angel of death, and then there's also other Amanita species, but they usually grow as lone mushrooms with a veil that's on them, and definitely never touch any of those. But there are many other fungi that can be dangerous as well. So my usual rule is just not to touch or eat anything that I find or I'm unsure about. I just take photos and try my best at identifying them. And honestly, that's still really fun. It's like a fun game of I Spy out in the forest, because it's not nearly as easy to spot mushrooms as it is to spot some of the larger plants.
Smike says they were shaped like that but didn't seem to group together that closely. Thank you for reminding me of this childhood memory. And,
great. You should go out and try to find some.
And Golden says,
those are fall mushrooms, though. That's like an September, October kind of mushroom.
And Golganak says, colloquially, that species, as kids, were called the puff balls. So is there also a species called puffball?
Yeah, so I mean, again, that's a common name, but when most people say the word puffball, they refer to, I should have one on there. They're no, I think scroll up for, yeah, there you go. They're usually larger and they're white. So, the average one you find might be about the size of maybe like a softball or a cantaloupe, but you can find massive ones if you're lucky that can be, soccer ball up to like a boulder size if they're really thriving. Very rare to find one because they are delicious, and many even amateur mycologists will like to find and consume them. But again, I do not recommend that. But even just seeing them can be really cool. I found some the size of my head before that were a bit past their prime, but they were still really interesting to see.
Dr. Waleed has a question for you. What's the consensus on between individual communication or interactions between fungi through their mycelium?
Yeah, so I think this might be referring to sort of like the Wood Wide Web, which is this thought that trees communicate via mycelia and fungi that associate with the roots of plants. This was thought to be the case for many years due to misinterpretation of a few studies. The current consent, and actually I've even I misunderstood this as well before kind of being a bit more integrated into the mycology community. But the current consensus is that is not how that the trees can't explicitly communicate between fungi that grow within their roots. But fungi, like of a given organism, certainly use their mycelia to spread nutrients, absorb water, secrete enzymes into the environment, and then absorb those nutrients, which is kind of how mushrooms eat per se. So they work akin to a plant root, but not exactly for a given fungal species, if that hopefully answers your question.
Nice. I again, the connection with sci-fi and science, whenever now mycelial network comes in, I don't know if you're a Star Trek fan, but that's always like the new Star Trek show. I'm like, oh, the mycelial spore network, and how does that work? Like, it's the immediate kicker coming on in.
Yeah, exactly. Yeah.
Smikes mentions it's you're producing the Canadian PSA about not putting foods in your mouth, so not eating these mushrooms. Laura Gale found ones. Remember like that, the lycoperdon.
Yep.
Pyriform.
That's it. Lycoperdon.
Yep. All right.
Yep.
Here we go. See, y'all know these fancy mycelial terms. I'm just like, ooh, I don't know. I'm like, I know Drosophila. It's about the extent of my fancy.
Yeah, that's the puffball. That's the common, or not the scientific name for the puffball.
Which one of these others are, favorites of yours? I mean, like, I like the Smurf Hut a lot.
Oh, okay. So that one on the left-hand side there, that's actually a chocolate slime mold. So that's technically not a fungus, but I wanted to put it on here because it looks exactly like a flaky chocolate bar. Let me see if I can pull it up. Or actually, maybe you can, if you look up chocolate slime mold, maybe on Google, we can get a zoomed in picture of it. It is the coolest slime mold I've ever seen. And again, this was just in like a local Ontario forest. And they grow kind of in these, see that one in the middle there? Yeah, exactly.
Oh, wow.
So they kind of grow in these little groupings of what looks like kind of flaky chocolate. You cannot eat them, but they are really, really cool to see in person.
It looks a little bit like our aspergillus, almost, like with the strands coming off. Do you find that there's similar structural features between these fungi out in nature, or are we just selectively looking at them right now?
Yeah, it's a great question. So I would say yes and no. There's actually more diversity in the fungal kingdom than even in the plant kingdom. And I think that's something that's often overlooked because the assumption is that all mushrooms are kind of just like the brown mushrooms we find in the grocery store, but there's huge phenotypic diversity within the fungal kingdom. I think that I certainly don't have a representative collection of all possible fungi within the Southwestern Ontario region. So probably it's just a little bit of kind of selection bias here, but definitely you can see patterns emerge between between structures in fungi, just like, for example, trees, many trees have leaves, but that doesn't always mean the leaves are exactly the same, but their function and somewhat of their shape are similar between trees, if that makes sense.
Yeah, absolutely. No, these are really cool, and they they totally look like they're strands of chocolate. And now
Yeah. Oh, yeah.
I can't not see it now. But
Yeah.
don't eat them. That's
No,
none of these
definitely not.
mushrooms.
Another cool one that I might have on there is also wolf's milk, which is another slime mold, and they form these perfect little pink balls. And they're very common in the fall out in the wild. I see them all. Once you've seen them once, you will see them everywhere. Yeah, and they grow as perfect little pink spheres, often on dead logs. They're really, really cool looking. But again, another slime mold, which is really cool because looking at them, and me as well, at first you think that they're going to be a fungus, and then actually with the help of iNaturalist, that was how I learned that this was not a fungus, it was a slime mold.
That is really cool that the program's able to delineate that much. And I know that they
Totally.
do the edge of life, where when you're tracking a particular organism, like as high north as you go, that you've identified it as far south, and that's like the range, latitude wise, of where this organism exists. So it's amazing that, again, that the app is something that this is possible, and you can do some really big citizen science on that front.
Totally. And it makes sense because as a scientist, I might want to know where people are spotting wolf's milk, but I can't go out into every single forest in Canada and picture it. So it's a really great way to source information and get a sense of even just trends of things that are happening.
Absolutely. And so, like, with your images, like, it's a beautiful collection. Do you know if there's any university, from the question on the chat, that has like a really nice collection that we can point people towards? For example, Smikes is right, at Harvard, they have a really giant ant collection because E.O. Wilson worked there for a long time.
Yeah.
Do you know if there is some kind of resource that we can provide to people to go to if they want to see, like, just a huge array of these samples?
Yeah, that's a really good question. And the short answer will be that I am not an expert in fungal diversity, so I might not have the correct answer here. I have been to the Harvard collection, and of course they also have they have collections of most ecological specimens that you can imagine. So if that is accessible to you, then that could be a good place to start. I know that there is a famous collection in the Netherlands. I would probably need to double check the name of that and its pronunciation, but I know that there's a collection of fungi in the Netherlands that is continuing to be developed. And I think those are the two that I would be most aware of off the top of my head. So unfortunately, I can't provide a great answer here, but if it's something you're really interested and passionate about, reach out to me on Twitter and I can do some research after this and try to find a better answer of where might be a good place to start.
By the way, folks, if you're interested in following Jessie on Twitter, there is her Twitter page right there. See, I got this. I'm ready to own it.
Yeah, you can see I mostly actually post pictures of fungi. So you can see one of my most recent tweets if you...
This is your most recent paper.
Yes, that's...
We'll have to ask you about this as well. We just getting questions, more questions. Waleed's wondering.
Yeah. Oh, there we go. Laura Gale has got it, and I don't want to mispronounce it, but yes, this institute in the Netherlands is the one I was referring to. So if you can see it in the chat, I don't want to mispronounce it, but it is that Fungal Biodiversity Institute, and that I think would be a great place to start if it's something that you are interested in. And actually, Amsterdam itself also has what they call something along the lines of the microbial zoo. It's this giant collection of microbes, and that includes both fungi and bacteria. So if you ever find yourself in Amsterdam and this is something that you're interested in, you could potentially check out both, would be my recommendation.
Perfect. Thank you, Laura Gale. Waleed was wondering, what are your thoughts on recent mushroom-based beverages?
Oh, well, let me open this link before I comment on something that I have not looked at. Let's see. Oh, I see. Okay. So I'll start by saying, prior to opening this link, I had not heard of this drink company. But I have heard of, there's also a coffee company that puts mushrooms in their coffee grounds. I think it's called 4-sigmatic, 4 as in the number. And I think that for me, with a lot of these products, again, I'm not a clinician, so I can't really comment on their use or if they provide anyone benefit. But I do know that in Canada, when it comes to supplements, there's not the same degree of regulation or requirement for meeting clinical indications as there is with, for example, an approved drug for clinical use. And so for me, I always use a healthy dose of skepticism when it comes to any type of supplement, because there's so little regulation around how much of something needs to go into something for it to be considered significant, the research that needs to be linked to the benefits that are being marketed. And that's a big issue with supplements in general, and kind of mushrooms fall into that realm. So there's a long way of saying that I don't really have anything against it. If people consume these products and like it and find that they have a benefit, that's totally fine. I guess I can say that I don't use any of them, and that's sort of my choice. I prefer to just eat as many mushrooms as I can when I come across them. But I really don't have anything against any of these products, but I'm always weary with supplements, mostly because I think they charge an arm and a leg for something that they don't have to show has the clinical benefit that they are advertising, if I can say that in as nice of a way as possible. So if you've tried them and like it, I'd be curious to hear about it, but I haven't tried them, and that's my 2 cents on supplements. So
yeah, so the Waleed was saying, I'm asking because I think because they make claims about certain common mushrooms used in beverages that would indicate some scientific research, but it's not clear whether or not, because I know some of these are broad claims, right? And it's who it's evaluated by, and
it's often related. So oftentimes a lot of those studies will reference, for example, animal studies, or potentially, maybe not the best designed sort of observational clinical studies. It can really vary all over the map in terms of the level of clinical research that is supporting the findings. And I would say when I have dug deep, me personally, I have not been satisfied with the benefit that is being advertised. But I'm also not a clinician, and I don't work for any of these companies. So if you are really interested in the effects or the potential benefits, I would just talk to your family doctor, and they would probably have a much better idea of the degree of research that really supports any clinical benefit of these types of compounds. The good news is that usually they then just don't have any activity, so all you're doing is wasting money. I don't think there's that many examples of things having a negative effect, but it's more that will you actually get the full benefit that's being advertised? Unknown. But again, I'm not a clinician, so reach out to your family doctor. Yeah.
We had a question from Smikes asking, what is the most common way to check for a new species, like the fungi that you're identifying is the genome? And then is that like a whole genome sequence, or are you looking at particular regions? So like if you're out in the field, and I know we're no ecologist of the mushrooms, but is that something that you would do if, or even like, I guess with the Canada strains, right? How would you identify that it's in fact that same one versus a different species?
Yeah, that's a really good question. So I wish I was an ecologist, but I am not. And for me personally, I would definitely start with a whole genome sequence. And there are research methods that exist to help you look at different genomes and determine how closely they are related in terms of if you have a different strain of a species or an entirely new species. So that would be where I would start, but there could be other strategies to identifying a novel species. But I think most people generally start with genomes these days.
Cliff Alistair McLean was wondering, are the same fungi generally spread all around the world in a certain climate zone, or are there many regional restricted species and subspecies?
Yeah, that's a great question, and a really, really broad one. So it honestly depends on the type of fungi that you're talking about. There are certainly fungi that exist, for example, both in North America and in Europe. And there are also ones that are much more regionally restricted. And I think it comes down to the types of things that affect any speciation, the environment, that they're growing in, what they need to grow. And I think all of that is definitely shifting with climate change. So even things that might have been restricted to a certain zone could be changing and things like that. But it's a great question. And unfortunately, the unsatisfying answer is that I think it's all over the map, and it really depends on what mushroom you're considering.
I mean, sometimes those are the best answers, right? It just means we need to look more to learn and figure it out.
Yeah.
Speaking of learning and figuring it out, this is your most recent paper, and we're just curious if you wanted to tell us about some of the hard hitting highlights of what your discovery was in this study, and what was the most challenging aspect of this study that you're the lead on. You're the is this like the, and also is this your big PhD paper? Is this just tell us a little bit about it.
Yeah, so this is one of them. This is the project that I've been talking a little bit about, or at least the initial portion of that project. So maybe we can just break down the title. That's probably the easiest thing that we can do. So in this paper, what I showed is this small molecule, right? This chemical that's made by bacteria. And those bacteria were these lactobacillus species. And you might recognize that name because that's the kind of bacteria that are usually considered probiotics. So you might find lactobacillus in yogurt, in supplements, in kefir, in any type of dairy product. And so that's when you might commonly come across lactobacillus. But because of that, they're also a major microbe that's already living in and inside of our body. So they're in our mouth, they're in our GI tract, they're all over within us. And so I found this chemical that's made by these lactobacillus that can inhibit the ability of Candida albicans, this fungus I've been talking about, to cause disease. And to do that, it actually specifically inhibits the virulence of the pathogen, which is something that I think is really cool and a big focus of my PhD, which has been that what if instead of killing a microbe, we can simply stop it from causing disease? And the reason behind that, as Belinda has talked a little bit about, is that a big issue with infectious disease is antimicrobial resistance. So these pathogens will develop resistance against our best drugs, and then suddenly we can't use them to treat disease anymore. And the reason that happens is there's this really big selective pressure, because you've taken this chemical that can come and just obliterate an entire population. And that creates this selective pressure, meaning that any bacteria or pathogen that can somehow manage to survive that massive extinction event will then be selected for. So if there's a random mutation in a bacteria that lets it survive in the presence of this drug, then it's going to be golden and it's going to proliferate and create this resistant infection. So what I wanted to do was try to think of a way that we could treat disease and stop this infection, but maybe not have that crazy selective pressure that leads to resistant disease. And to do that, what we can do is specifically target the virulence of these pathogens. So what are they actually doing to our body to cause disease? And can we inhibit that as opposed to just outright killing all the fungus that's around us? And to do that, that's this other word in the title, filamentation. This is this really important virulence trait in fungi that lets them cause disease in our bodies. And that's because what they can do is basically transition between like a yeast-like ball form that they're growing in into kind of this worm-like structure that they can use to bind to and then invade into our tissues to cause disease. So if we can stop that from happening, then we can potentially prevent the infection without actually killing the fungus and selecting for resistance. And basically this molecule that I discovered that's made by bacteria can specifically do that, because that's what's happening in our bodies, at least potentially. Still obviously working on this research, but potentially the lactobacillus are specifically making this compound so that they can stop the fungus from causing disease. As one example, one hypothesis might be if they cause disease, it's going to recruit a bunch of immune cells, and they could also affect the bacteria. So it might be in the bacteria's best interest to stop that from happening. And that's sort of the breakdown of the title. The last part there is just how I figured out how this is actually happening, what the chemical actually does to the fungus to stop disease.
Yeah, I like the title breakdown a lot because I feel like a lot of scientific papers almost read like a Harry Potter title, right? Harry
Yeah.
Potter and just
Yeah.
a bunch of words thrown in. What is
And
the?
it's actually done on purpose because, like, as a researcher, when I want to find papers in my field, I'll often choose the most specific words possible to get those papers. So then, in accordance with that, researchers will then make their title as specific as possible so people in this exact field can find the paper and then read it. So it's like a catch-22.
Yeah, and Smikes is a little bit like a gatekeeping feel almost.
Oh, totally. Yeah. And it's actually really frustrating because this was one of the big issues with the COVID-19 pandemic. There was all kinds of research being published, and then it was either behind a paywall or in complete scientific jargon. I think what makes me really upset with scientific communication is people talk about, communicating to the public. It's not that the public doesn't understand science. It's that scientists use this incredibly jargon-filled language that makes it almost impossible to understand what they're talking about, even though conceptually what's going on, anyone can understand. But it's like you're speaking a foreign language that people just don't have, are just not fluent in that language. It's not actually that, the public is not understanding science. It's that we are speaking in a language that most people don't speak.
And speaking of languages that most people don't speak, that's something that you've rallied against. There was a really cool video from a soapbox science challenge that you did. And Dacum, welcome on in. This is our guest, again, Jessie MacAlpine. All her info is linked at the top. It is pinned, so you can check out her website. So one of the things that you did was this challenge, right, to make that science accessible. And especially, so the video was in 2021. So we're still in like the midst of the COVID pandemic. And, some would argue we still are. How do you use your knowledge and your platform to inform people, like non-scientists, these scientific principles? And like, what was that soapbox challenge? And for those interested, I'll link the video right now into the chat, and we'll also link it on the YouTube video coming out later.
Yeah, so Soapbox Science is really interesting. It's an international organization, and what they do is basically connect researchers, usually with some sort of outdoor festival where they can communicate their science. So unfortunately, because I participated in 2021, it was an entirely virtual event, which means you can watch it on YouTube. In previous years, and I think what's now on or has now resumed, is that usually this actually takes place outside on a literal soapbox where you use kind of props and things like that to sort of be like, hey, are you interested in, whatever kind of the catchy one sentence saying you can use to get people interested in your work. So I usually say like, oh, are you interested in the rainforest that's living inside of your body? Because that's one analogy that we can use to describe this community of microbes that's living inside of us. It's as dense and, or I should say, a comparison is that it could be as dense and as diverse as the rainforest community. But we don't often think about when we think about this kind of community of bacteria and fungi living within our body. And that was kind of my launch to then say, what could be living within this rainforest? What might we not be seeing that we can then harness to combat some of these infections and diseases that we're struggling to treat with resistance emerging.
And who's the audience for these soapbox
public
challenges?
So totally public. Because it's usually on the street, it's usually just whoever happens to walk past that day. I think because it was a virtual event, it ended up being a little bit more international, but maybe a little bit more closer to home in the academia front, just because that's who ended up being I think reached out to the most through the academic networks. But usually it just takes place, 1 year it was right in downtown Toronto, in one of the busiest intersections in Toronto, and it was just whoever happened to walk by trying to get them interested in chatting about science. So I was a little bit disappointed I couldn't participate in that element of it, but obviously that was not safe in 2021, so it was still a really fun challenge to do that virtually as well.
That sounds like, frontline SciComm happening right there. You're just like in the
Yeah.
streets, just telling people. And I really like
Yeah.
that analogy. And actually, all the analogies that you've presented us today are so accessible.
Oh,
How do you wrap your head around, like, what did you do to make science so accessible, like in how you're explaining things? Because you've never today explained things using jargon. It was all, like, I think pretty readily accessible to many folks in the chat as we're getting questions, and everyone's pretty, at least in my mind, is following along. Like, what do you do to prep yourself to explain these kinds of concepts, and how do you come up with these analogies? Because I feel like many scientists could appreciate that kind of advice or help that we don't readily have.
Yeah, I think for me, when it comes to analogies, I think there's two things. One, I think growing up in the science fair system meant that from day one of doing research, I had to be able to explain my project to the science fair judges, who usually came from pretty much any walk of life. So I might be doing a biology project, but presenting to an engineer and needing to communicate to them the level and rigor of research that I'd done, but obviously not knowing that they did not have technical expertise in molecular biology, as an example. When I try to think about analogies, I think one thing that's really important, and this is going to sound a little bit funny, is that to not focus on the most perfect analogy. So if we use the gut rainforest as an example, obviously there's not a rainforest living in our gut. But I'm just trying to think of something that most people will have a reasonable grasp on. Most people are aware of, how dense, how rich a rainforest is. And that's what I want people to picture when they picture our microbiome. But I don't want people to get stuck in the nitty gritties of a rainforest has, and I'm going to just make up numbers here, but let's say a rainforest has 25,000 species and our gut has 28,000 species, or something like that. Those are not accurate numbers. I'm just using that for an example here. So I think where I try to focus on it is something that's accessible and basically provides the mental image of what I am trying to deliver with the science that I'm talking about, if that makes sense.
Yeah, it absolutely does. And Dakum in chat says it's nice to not have to have dictionaries and things while listening to you and understanding.
That's great, thank you.
That's why it's, again, I think, been a really great talk, and like kind of getting in your mind of how you're so passionate about science communication, but it's not this I'm pretending to be passionate. It's like, clearly you really care, and you've thought out like how you explain these concepts, and I think that makes a world of a difference to scientists and non-scientists alike. So as we start to wrap up, Jessie, what are you said your next step is to go be a postdoctoral researcher at the National Institute of Health. What are your long-term goals as a scientist, and where, the silly standard question of where do you see yourself in a decade, right? Like what is your ideal world where you're doing the science communication, presumably keeping that up? Like where do you see yourself?
Yeah, so this is easier said than done, and I know that this will live on YouTube in perpetuity, so we'll see where this goes. But at the moment, my dream is to stay in academia. So I would like to be, like a professor, for lack of a better word, someone who's running an academic research lab and then also teaching at a research intensive university, hopefully back in Canada. But again, it's a tough field with few jobs, so it's much easier said than done. But I definitely think one thing I can say for sure is that no matter where I end up, I want to still be doing research, as that's what's always driven me and made me the most excited since truly I was, a young kid. And I always want to be communicating that science. I think that it's a service to the community, that if you are discovering all this cool stuff, then it's our job to help share that with people. And I think that is maybe what's a misconception about science, is that no one is more excited than a scientist about the most like oblique, obscure, really specific area of maybe like some specific element of biology. So it's not hard to find the passion. I think it can be maybe always translating that to the public is where we sometimes get stuck, because it's unfortunately not always prioritized with jobs and funding and things like that. So maybe in the future, one thing that could be great is kind of pushing that to be a little bit more of a priority as someone who's an academic, as kind of like, we're the experts. It's our jobs to share all this cool science. So yeah, hopefully I will be able to continue to do research and continue to talk about science in whatever capacity that may be. That's pretty much the dream. So
I love it. Few more questions just to further demystify the science, and if anyone in the chat has any questions for Jessie, please feel free to post them in the chat. We don't want to take up too much of her time, but we'll take a little bit more. One thing that is regularly asked is, what is your everyday life look like in a lab? We see people on TV in a lab coat and a pipette tip, and they're mixing colored chemicals and sequence this genome, and it's done instantaneously. But what, as a fungal geneticist, what is your day-to-day like?
Yeah, so I think I'll start by basically not answering the question, which is that what I love the most about science is that every day is actually pretty different. Some days I might be doing data analysis, some days I might be doing experiments, some days I might be lucky and be at a conference where I get to talk about my science. But I guess I would say generally the day-to-day is actually a little bit close to the TV science, where we do wear lab coats. I do literally use a Bunsen burner every single day. But usually what I'm doing is I grow fungus and then kind of find different ways to kill it, is maybe one sentence, and to study its biology. So a lot of the things I use most commonly in the lab are microscopes. I do a lot of microscopy. I look at the fungi. I see how they're growing, how they undergo this process that I was talking about, where they kind of elongate into these worm-like structures, and how that happens. I do a lot of genetics. So that means I grow a lot of fungi and then use different techniques to genetically manipulate them. So that might be to delete a gene and see what that does to the fungus. That might be to add something in and see what happens there. That's another common thing that I do every single day. And then, again, sort of looking at how we can kill the fungus. So testing different drugs, seeing how that affects their growth, seeing that how that might affect mammalian cells, as an example. And that's a lot of my sort of day-to-day work. But it really is me at a bench doing a lot of experiments. And there's a faculty member in our department who once said that you have to be really passionate about science, because grad school is pipetting clear liquids from one container into another. And for anyone who doesn't know what a pipette is, it's basically just this tool we can use to pick up really small volumes of liquid. And so the joke is that in grad school, you spend your whole time picking up small volumes of liquid from one tube and putting them into another. And unfortunately, that is the case a lot of the time. But what's cool is what you are moving and why. So even though physically from the outside it might not look that cool, usually it's still pretty cool what's actually happening kind of at the microscopic level.
I really wish the liquid was colored, though, so you can tell if you were getting any clues as to what you were finding.
Although color can have so much impact on biology, especially if you're doing microscopy. So we actually all love colorless liquid in the labs, but that's why you have to really carefully label everything, because all the colorless liquids look the same.
We have a question from 3D Prima. What's the name again of the fungus that crawls into other fungi, earthborn usually?
Crawls into other fungi. I might need a little bit more details here. Or are we talking about the fungi that that infects insects?
Yeah, so 3D Prima let us know. And while you're letting us know, they'd had a follow-up. Was there a great change in mycology with CRISPR-Cas9?
Yeah, great question. And the short answer is yes. If you were here earlier in the talk, I was mentioning that in the fungus that I study, it's really, really hard to do traditional genetics because you can't mate in the same way to make different genetic mutants. So when CRISPR-Cas9 came out, it really accelerated the speed with which we could generate mutants. And so now in the fungus that I study, we can much more rapidly study different genetic mutants. And now we have all of these cool genetic resources to be able to ask really specific questions about the biology of these organisms and how they cause disease. So it's honestly hard to pinpoint exactly how much of an impact it's had on the field, because it's really changed everyone's day-to-day by a long shot. So it's something that I have benefited immensely from. And actually, I was in the lab before we started doing CRISPR genetic manipulation, and now I've been since, and my life has also greatly improved.
Golganak has a potentially off-topic and likely basic question. Curious about mycoproteins, especially as a development as a meat substitute.
Yeah, it's a great question. So I also mentioned earlier in the presentation today about how fungi are more closely related to animals than to plants. And so in some ways that means their biology is a little bit different. And what you may know is, if you think about the five tastes, and umami is the fifth one, and that's a very common flavor profile that you can achieve with mushrooms related to some of this unique biology. And so I am personally a huge fan of meat alternatives, and I think that fungi are absolutely one of the best directions to head in. If you've heard of Beyond Meat and Beyond Burgers, one of the ways that they were able to achieve that meat-like flavor was actually using yeast that were genetically manipulated to produce heme. And I'm pretty sure if this is correct, someone please correct me if I'm wrong, but I remember reading this back in the day. And that kind of gives it that almost metallic taste that's closer to meat. So fungi are going to be really important for not only the bulk production that's needed to have success with meat alternatives, but I think also for the flavor profile to make it attractive to as broad of an audience as possible. I don't know if that answers it or if I'm just giving my opinion, but basically I think it's really cool. And I agree that, it's a rich, diverse source of protein, a way that we can still, have somewhat of a natural source of protein, and fungi grow really easily. So it's could hopefully have less of an impact on the environment in terms of scaled production as well.
So V's wondering yeast. So that's the main reason why Beyond and Impossible, to say the least, made their vegan taste like the meat.
Yes. So that is what I recall from an article that interviewed Mike Eisen, who's not Mike Eisen, but one of the creators of the Beyond and Impossible burgers. But yeah, one of the tricks, I think there's many things involved in the flavor, but one of the key tricks was yeast making heme, which is a component of it's a protein found in blood.
And then Jessie, just to round out, what are you most excited about this coming year?
Oh, this year? Well,
or just
in my personal life, or in general?
In your personal life, in science, like what are you really like career wise? Starting in the new lab. Like, what is it that you're really looking forward to most?
That's sort of it. Starting in the new lab, I'm really excited about. I can't talk too much about the project that I'm going to be doing, but I'm very excited about it. It's still in the realm of fungal biology, and basically I'm just really excited to get started. So I can't wait for that to begin.
Awesome. Well, best of luck. I'm sure you're going to crush it. Any last minute questions coming in? Is there anything you'd like to plug and share about you have any upcoming SciComm endeavors, any website info that you want to point us towards, anything at all? Now's your time.
Yeah, I think I don't have anything specific to point to on my end, but I think I just generally want to put out there that I've said it like at least 3 times, but I really do believe that science is a fundamental part of being human. And so even if you aren't a scientist or don't see yourself as a scientist, I really want to challenge you to challenge that narrative, because so much about being human is kind of doing science every day, like finding, deciding that your dinner doesn't taste right, adding a bit of salt, seeing, okay, that only helped a bit, adding something else. All that's sort of the process of experimentation. And I think I really want to encourage that even if you think science is not for you, or I'm not a scientist, it really can be. And there's so many cool things to explore out in the natural world. And hopefully that can help you continue to be open to just spreading the love of science, as I have tried to do here today.
Beautifully said. I have to ask this question. Do you believe that there are mushrooms on other planets?
Oh, mushrooms on... Well, okay, I guess I'm going to cheat a little bit here and say maybe not necessarily mushrooms, but fungi are at least known to be able to survive at some degree of extremes, as are bacteria. I can't specifically comment. This is not my area of expertise, but I can say my personal opinion is that definitely there are microbes out there. And I think it's only a matter of time before we find them, whether they are fungi or animal-like cells or plant-like cells, I guess, or something entirely new. I don't know, but I think that thinking of the scale of the universe and the stochasticity with which our life emerged, I think that there is absolutely life out there, and I hope it's fungi, but we'll see.
Well said. So I think on that we'll wrap on up. Jessie, thank you so much for your time today, for taking us, through your life journey and your love of science, everything from being on a television show to, working in the lab, and that love of the science fair and that big difference it made in your life. I think it's a really inspiring story, and thank you so much for taking the time to be here with us and chat with us in the inaugural, the first ever Genetic Society of America live stream on Twitch. We'll actually be back again, y'all, next week. Next Tuesday, we have the next iteration of this stream, so we'll hope you all join us then. It's a member of the accessibility subcommittee of the early career leadership program that we have to show off. Really quickly, just some announcement. If y'all liked what y'all saw today, there is the opportunity. We are a professional society that you're able to donate to the Genetic Society of America. You can donate to help sustain our career programs, such as the Early Career Leadership Program. All of that information is available on that website from Nightbot, not from Stream Elements. Again, with that, Jessie, thank you so much for your time. Thank you all for the viewers for being here and asking questions and being curious. We'll see y'all next Tuesday, same time, 3 p.m. Eastern Standard, for our next iteration, the Genetic Society of America, live on Twitch. Have a great one, folks. Bye, everyone.